Ms. Stefanie Countryman. Ms. Countryman is the Director of BioServe Space Technologies and a Research Associate within the Ann and H.G. Smead Aerospace Engineering and Science Department at the University of Colorado Boulder. As Director, she leads a wide variety of activities, including programmatic oversight of space life science investigations and engineering projects. Recently, she has been involved with experiments examining microgravity effects on proximal tubule epithelial cells of the kidney, induced pluripotent stem cells, mesenchymal stem cells, cardiomyocytes, and endothelial cells. Each of these experiments seeks to utilize the microgravity environment as a model for different types of human disease, including aging, in order to seek more effective treatments for people on Earth. Stefanie, please feel free to start when you're ready.
Thanks, Mark. So today I'm going to talk about going from Benchtop to the International Space Station. So essentially, how do we translate a ground-based experiment into a spaceflight experiment, and specifically utilizing one of the microphysiological systems platforms. Duck Ho did such a great job at talking about the engineered heart tissue platform that I'm going to switch gears and talk about one of the other platforms that we also supported. Of course, I could talk all day on this subject, but I'm going to try to cover top-level details in the next 20 minutes. Just quickly tell you a little bit about who BioServe is. We are one of what NASA and ISS National Lab considers one of the implementation partners. We were established at the University of Colorado in Boulder in 1987. We have about 20 to 25 full-time employees who consist of faculty, staff, engineers, and students. We have extensive experience launching life science experiments to space. We flew our first experiment in 1991 on board the Space Shuttle STS-37. Since that time, we've launched experiments on 72 spaceflight missions, and we've had facilities continuously on board the International Space Station since 2001. In just the last 2 years alone, we've supported 15 different complex space-based cell culture experiments, including microphysiological systems and organoids experiments. And by the end of this year, we have 4 more on the books. So I'm going to jump right into talking about translating experiments from the ground to space. This right here is just a picture of a typical cell culture lab or wet lab, labs that you'll see in lab facilities, pharmaceutical biotech companies, science labs, and universities around on Earth. And this is what our science lab looks like on board the ISS. This is the Destiny lab, and this is where the majority of the US experiments are conducted. And you can see that there's quite a bit of equipment in this lab, and a lot of this equipment does support different types of life science experiments. There's even equipment that looks just like it does on the ground. This is on the right side is a picture of Anne McLean using a microscope to do some imaging. In terms of equipment, here are some of the pieces of lab equipment that are necessary, that are utilized on Earth for conducting cell culture experiments. We have a biosafety cabinet, which is designed not only to do sterile work, but protect the scientist who's doing that work. We have incubators, 37C, 5% CO2. That's how we conduct our cell cultures. We use, with MPS platforms, we use syringe pumps, and this is what that may look like in the lab. We have -80 freezers, -20 freezers, 4C refrigeration. There's pipetting that occurs in the lab. Rapid freezing is done with liquid nitrogen. We have water baths and centrifuges. So these are all types of equipment that you may see in a standard lab. This is what this equipment looks like on board the ISS. So up here on the left, we have the equivalent to the biosafety cabinet on the ground. It's called the Life Sciences Glove Box. And this is designed to protect the crew and the vehicle from any organisms or liquids that could be dangerous to the crew and vehicle. But it also allows us an opportunity to use a space that is not necessarily completely sterile, but as sterile as we can get on board the space station. We have -80 freezers. It's called Melfi. This is the crew with the -80 freezer. These 4 round doors are the -80 freezer space. We do have, BioServe has an incubator up on station. It's called Sable. It's these 2 units right here. We currently have 3 of them on board. They provide the 37C and the active 5% CO2, but they can also provide a temperature range of -5C to +43. In terms of a lab bench, if you're doing non-sterile work, we have the multi-purpose work area right here. We have a thaw system, that water bath that you saw. Obviously, we can't have loose liquids on board the station. So we developed a thaw system that is equivalent to a water bath, but it's designed for use in a microgravity environment. There's also a microscope and imaging system that's right here. This is a crew conducting a protein crystallography experiment. There is a centrifuge on board that's primarily used for spinning blood from the astronauts, but it can also be used in cell culture experiments. Just recently, a rapid freeze piece of hardware was designed and launched to the space station, which will be the equivalent of, or similar to, utilizing liquid nitrogen. Up until just recently, this was not available. And then in terms of pipetting, if you're using liquids that aren't hazardous or small volumes where you can use surface tension to maintain those liquids, you can do standard pipetting. But at times when we have larger versions of pipetting, we have to use syringes in an enclosed loop system in order to do that. So how do we translate to the ISS? Well, there's lots of things to consider. So we can consider whether we want to do a manual experiment, which you saw some pictures in the previous slide of the crew conducting experiments manually, versus automated. And there's pros and cons to both types. The number of samples, sometimes with automated systems, because of our limited space, we can have a lower number of samples than the manual. Though, depending upon the processes and procedures that we use for those manual systems, that may also limit the number of samples that are available. Teams need to consider whether they want to launch their cells live or frozen. And that may be dependent upon whether your cells are adherent or suspended. Obviously, if you launch adherent cells, say in a cryovial, if you want to thaw them in orbit and then seed them in orbit in a cell culture plate, that can be tricky because there's no gravity to help those cells settle down to the bottom of the plate. So it's very likely that adherent cells will not adhere if seeded in microgravity. You also need to consider the number of days that it takes for your experiment to get to orbit and then be accessible. You have to think about launch effects, g-forces and vibration, your launch orientation, whether you have requirements for active CO2. You also need to think about thermal control and the uniformity of your thermal control. Automated systems often have electronics in them that generate heat, and so that can actually make hot spots within your system. So managing thermal control is very important. And then in terms of returning live or frozen or fixed, those are also things to consider. And essentially you have the same things. You have to consider CO2 if you're coming back live, and thermal control, and then the landing effects on your live cultures, or even some of your fixed cultures if you're looking at morphology. So you want to consider launch orientation, and then of course the number of days until access to those samples, since it can be a few days from when the capsule unbursts with the station to when it lands in the ocean. You also have to consider bubbles and bubble management. Obviously, the bubbles don't rise in space, and so they can sit right in the middle of your culture, and that can impact either your fluidics or the morphology of your cells growing. And then the temperature control of your samples and medium reagents as they're being transported and returned from the station. You have to consider human factors, how the crew will follow procedures to either power up your powered payload or to complete your manual operations. There's obviously power issues and concerns that you need to address, and then of course material compatibility, gas permeability, and sterility with the unit. And also you need to consider processes that you do in the lab that actually utilize gravity. How do you translate them to the spaceflight experiment? So I'm going to talk quickly about a specific example of an MPS device, which is the Nordis Bio triplex chip, organ on a chip, that we designed a syringe pump system for and launched to the station on SpaceX 17. So this experiment was called the effect of microgravity on the structure and function of proximal and distal tubule in a microphysiological system. The PIs were Jonathan Himmelfarb, Ed Kelly, and Catherine Young at the University of Washington, and it was sponsored by the NIH, NCATS, ISS National Lab, and NASA. So the requirements for this experiment, on the right here, you can see the Nordis triplex chip. So each chip had 3 samples on it, and each chip had both an input, and an output. There were 24 chips that the team wanted to launch, so that was a total of 72 samples. The experiment required 37 C, 5% CO2, and high humidity to keep the samples from drying out, since these chips have very low volumes of fluid. They did need a correct launch orientation. The cells were loaded in, the cells formed little tubules in the chips, and they needed those tubules to maintain their shape during launch. So we did have to consider launch orientation. The media that was not used during the transport of the system needed to be held at 4C. The system required continuous perfusion of the media from when they were loaded into the system until the samples were terminated. And that perfusion rate was required at 750 microliters a day. There also was a requirement to collect the effluent from each individual sample and then store that effluent at -80. And then at termination, the samples were either fixed or preserved. The perfusion rate of the fixative and preservation reagent had a different rate of 1,440 microliters a day. So the experiment needed 7 days of microgravity exposure, and it required 2 days of microgravity exposure for the treatment media. It had acclimation media and treatment media, and it also required 4 to 24 hours of fixation. So what are the things that we needed to adapt for the spaceflight system? Well, there were 4 different use configurations. There was the launch configuration, the on-orbit configuration, both inside the stable incubator and inside the life sciences glove box. Because this was a semi-automated system, it required some input by the crew. And then there also was a return scenario. So as we said, we continually perfused the samples until they were terminated. We needed temperature control through all of these phases of the experiment. We also needed adequate 5% CO2. So we did have static CO2 during the launch, and then once it was inserted into the stable incubator, it was active. The treatment media was kept at 4C both on launch and during on-orbit stowage until use. The fixed samples themselves were stored at 4C on orbit and then kept at 4C during return. The preserved samples were kept at -80 C on orbit and then returned at -80. We also had to consider the NASA required levels of containment. So for the cells in the effluent, that required 2 levels of containment to meet NASA safety guidelines, but that containment needed to allow for the appropriate gas exchange so that the cultures could get the required CO2. The formalin cassettes needed 3 levels of containment because that's considered a hazard level 2 from NASA, which requires 3 levels of containment. And again, as I mentioned, we need to think about thermal control throughout all phases, and then the human factors. One of the other things that we had to consider was the material biocompatibility. So it's very important for the materials of these systems like this not to impact the science. So in fact, every single piece of material that came in contact with the science, we sent to the PI, and then they did testing in Petri dishes with their cells to ensure that those pieces of material did not impact the science. That material also has to meet NASA safety requirements in that it cannot have significant off-gassing or affect the crew in any way. The material obviously needs to be able to be sterilized, and it also needed to be able to handle -80 C to +37 C. The whole system itself, in addition to the science, needed to withstand launch vibrations and G-forces since we were launching live cells. And then once the system was on board ISS, we needed to have real-time telemetry to ensure that the system was working correctly. But we also needed to record all other times that the system was not inside stable so that we could know that the parameters were within normal and that we didn't have any issues outside of the time that it was inside the incubator. So all of this needed to fit within a compact volume. So we had one launch locker to fit all of this, the 72 samples and all the supporting hardware for that. And then we had 2 stables on orbit. So for visual sakes, this is what, if we had 39 samples in the lab, this is what it takes to support that using a syringe pump system. So 72 samples would require 8 syringe pumps on the ground, 4 incubators, 24 large petri dishes to hold those triplex chips so that they did not become contaminated, 216 small petri dishes for all of the effluent that would be collected, 216 syringes for media and fixative, and 216 feet of tubing. Just so everyone knows, the Nordis BioChip system does have a pneumatic system as well that takes up a little bit less space, but it still has quite a high volume. So our challenge was to take all of this, meet all of those requirements, and get it into a system that would now fit into the space flight parameters that we had. So visually, we needed to take 48 cubic feet that we used to support this experiment in the lab. And for launch, we had 2 cubic feet. And then on orbit, we had 1.6 cubic feet. So how did we do that? Well, we did a significant amount of research and development, obviously, in determining how to develop this system. There was a significant amount of testing that was completed both on the engineering side and the science side, and then the 2 sides working together to test the system in the engineering system to ensure that the design was supporting the science appropriately. In the end, the whole system and all of the supporting hardware required to support the protocol, we machined 2,500 individual pieces of hardware. So to show you quickly, the housing, so for the triplex chip, because it contained human cells, we needed to put that in a level of containment, 2 levels of containment. So we developed a housing. On the left picture, you see the square box and a valve block, which is in the right hand of the person in this left picture. And the 3 chips, each housing held 3 chips. And this housing was completely sealed. And on the right, you can see it's fully loaded, this housing. And this housing and valve block system would then interface with the media and fixative cassette. So here we have the media and fixative cassettes integrated with the housing. So the housing provided, it was 2 levels of containment. It was able to provide gas exchange for the cell cultures, but it kept all of the liquids contained if there was any leaks. And then the media cassettes, which had 9 channels, because we were supporting, each housing was supporting 9 samples, 9 channels of media, and then 9 channels, 9 individual bags collecting the effluent from each individual sample. This is the syringe pump itself. So 2 housings with their associated media cassette would fit within the syringe pump, and the syringe pump could purge, it could run. The run was when we were doing media at 750 microliters a day. The fixative was the 1440 microliters a day, and then of course we needed to retract the pistons that you see here in order to pull the samples out of here when the crew needed to do a media exchange. So just to remind you, this is what it looked like in the lab. And in the end, this is what it looked like, but we had 4 of these. So we were able to reduce that down to 4 of these. One of the syringe pumps, and then there's 4 loaded within the launch locker, and then it's sealed up and powered and ready to launch while it's on the Dragon and its capsule, and it's being powered continuously through this entire transport. This is what it looked like on orbit. So here are the 2 stable units. Here is the inside of the stable units. There were 2 syringe pumps and 1 power module inside each stable unit. So for a total of 4 syringe pumps and 2 power modules. And then this is what it looked like inside of the LSG when the crew was getting ready to pull these out to do a media exchange. The LSG could hold 2 of these syringe pump systems at a time.
It's Stephanie.
And then this, yep.
Sorry to interrupt. I'm going to ask that we potentially, okay, now your slides are moving forward. So we see your launch configuration now. So, okay, please proceed.
So I'm
okay,
behind, where am I? Am I on
on
return configuration?
You're on return configuration now. Thank you.
Okay, did everybody see this one?
Yep, that's coming through now. And now
Okay,
Onboard configuration.
Okay, so I don't know if people saw this. So so I'm not sure what everybody saw. So this was the on-orbit stable configuration, and this was the LSG on-orbit configuration. And then this was the return configuration where we only brought the cassettes and the effluent cassettes, and then of course the the culture samples back. Some were brought back at 4C and some were brought back at -80. So the challenge is to supporting any type of experiment that's complex like this, that's a microphysiological system platform or organoids even, is reducing to a small footprint, understanding fluid flow and management, understanding fluid flow and managing that fluid flow, and particularly with regards to bubbles. It's challenging to create the appropriate levels of containment, but keep gas permeability at a level that is amenable to your cell cultures. Obviously, we use quite a few plastic components. A lot of plastics are very biocompatible, but machining those plastic parts is very complex, and the tolerances on our plastic parts were very small, and so that was very challenging. Obviously, getting the correct interface between the chips and the cassettes so that you could feed the appropriate media to the chips. Loading quickly and efficiently, designing a system that you can load quickly and efficiently with the science minus bubbles, but keeping appropriate temperature control is very challenging. Keeping sterility during all of the loading and handling, managing the effluent, and keeping everything uniform, and obviously designing a circuit board that is conducive to small spaces. These are all challenges that are present when you're trying to translate experiments from the ground-based to the space-based, but can be managed and met successfully. So my words of wisdom in all of this is that, as I said, it's challenging, yet you can have great success if you plan appropriately. I would say that you need to plan for an adequate timeline. There are times when rapid timelines force innovation, but also due to the complexity of translating from ground-based to space-based, it can also make room for error if you go too fast. I would say that your science and engineering teams need to work really closely together so that the results that you get with your science are due to microgravity rather than a hardware configuration. I would say, I think as Jean talked about, try to plan for multiple flights. It's hard to get things done 100% correctly on the first flight. So if you can have multiple flights, you can address issues and concerns and on subsequent flights. And then of course, you can never do too little or you can never do too much testing. It's very important in this business to test as much as possible, both the hardware and the science, and then the science and hardware together. So that's the end of my talk. Thank you. I'd like to recognize again the NIH NCATS, ISS National Lab, Nordis Bio, the UW, and then of course all of the science teams that we work with. Thank you.